Antibodies were discovered in 1890 by physiologist Emil von Behring and microbiologist Shibasaburo Kitasato as protective antitoxins in the blood of animals exposed to diphtheria or tetanus toxin. Ever since their discovery, antibodies have been used as a basis for serum therapy, the treatment of infectious diseases using blood serum from immunized animals, and as diagnostic tools to monitor infectious disease. Antibodies play a critical role in the adaptive immune system, which can fight off an infection with high specificity. When a foreign object or pathogen enters the body, the receptors on the surface of a B cell can recognize and bind to specific parts of a pathogen called antigens. Once B cells bind to antigens, these B cells can differentiate into plasma cells and produce antibodies, which are released into the bloodstream. There, antibodies can bind to the antigen on the virus or bacteria, helping to fight the infection through neutralization, complement recruitment, and opsonization. Each B cell has a specific type of antibody, resulting in specific immune responses against invading pathogens. However, antibodies specific for any given antigen came as mixtures of antibodies, produced by thousands of antibody-producing cells in an immunized animal. Each of these cells produced an antibody of its own kind, so that antibody specificity was a property of the antibody population rather than those of individual antibodies. Scientists wondered if it was possible to produce monoclonal antibodies or homogeneous antibodies specific to a single antigen. Monoclonal antibodies are much more useful compared to polyclonal antibodies because they can be used for antigenic characterization of pathogens. Monoclonal antibodies would be composed of identical antibodies that can specifically target a single antigen, whereas polyclonal antibodies are made of a group of different antibodies, each of which are specific to different antigens. In 1975, immunologists Georges Koller and Cesar Milston conducted an experiment that described the production of specific monoclonal antibodies. They had taken the spleen cell from a mouse that was injected with sheep red blood cells. That way, the spleen cells would produce antibodies that would specifically target the sheep red blood cells. However, the spleen cells had a limited lifespan, so they decided to fuse myeloma cells with the antibody-producing spleen cells. Myeloma cells are cancer cells, which were very accessible at the time. Because of their accessibility, they were also well researched. These tumor cells could also survive for long periods of time. By fusing these cells with the now acquired spleen cells, they could then create new cells that were immortal outside of the body. The fusion of these cells, specifically spleen cells that created antibodies and myeloma cells, are called hybridomas. These cells produced monoclonal antibodies. However, there was a problem. The problem was that the hybridomas were creating two types of antibodies: the desired antibody, produced by the spleen cells, and a non-specific antibody, produced by the myeloma cells. This issue was then fixed by using a myeloma cell that produced no antibody. To isolate the hybridoma cells that secreted antibodies specific for sheep blood, they spread sheep red blood cells on agar plates. On specific spaced-out locations, they placed the hybridoma cells. Within time, the hybridoma cells that secreted antibodies specific for sheep blood cleared up sheep blood around the cell colony, producing an area of clearance. This helped them distinguish between the antibodies that were specific and those that were not. The discovery of monoclonal antibodies was innovative, leading to potential treatments for virulent viruses. New cures have made significant progress in decreasing deaths, hospitalization, and creating a barrier safeguarding humanity from an unexpected wave of diseases. One prominent example of using monoclonal antibodies is for the SARS-CoV-2 virus, commonly known as the COVID-19. In order to fight against the coronavirus pandemic, scientists have developed an anti-COVID treatment using monoclonal antibodies. To make this, the scientists had to trace back to a survivor of the SARS-CoV-2 virus from 2003. The immune cells from the individual were isolated and used to produce hybridoma cells that could mass-produce coronavirus-specific antibodies. These antibodies can then be injected into an infected individual, reducing the potential risk of death due to COVID and decreasing hospitalization. Another recognizable use of monoclonal antibodies is in cancer prevention. Monoclonal antibodies can target cancer cells that attack the immune system and kill them and prevent them from growth. Some cancer-targeting monoclonal antibodies detect specific proteins in cancer cells and block signals or receptors to stop cancer cells from duplicating. They also help the immune system fight against cancer. Another use of monoclonal antibodies is in treating Alzheimer's disease. An antibody called lecanemab is considered as a possibility to treat the disease by dissolving amyloid plaques in the brain.